Welcome to another event of our Translational Neuroimaging Educational Program. In this program, we host a series of talks and presentations aiming to engage the neuroimaging and brain science community to enhance the translational application of computational neuroimaging techniques in promotion of human brain health. This educational program is nested within the Lifespan Working Group of the ENIGMA Consortium. This is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The hope here is really to provide opportunities to expand skills, initiate collaborations, and facilitate networking in a welcoming learning environment. Here's just Dr. Yanghee and myself, who are the directors and put this program together, along with our core advisory committee listed here. And if you're interested in learning more on the website itself, you can enter your email address and you'll receive updates there. But otherwise, all the sessions that we've previously had are available on our YouTube channel, which you can see linked here. And yeah, all of the past events are available to refer back to there. And you can also follow us on X or Blue Sky to learn more about future events. If you're interested in showcasing some of your own work or methods on this platform, please don't hesitate to reach out to Young and myself. We'd be happy to discuss whether the platform would be a good place for showcasing some of the work you've done. But today it is my pleasure to introduce Yanghee Im. Yanghee is a neuroscience PhD student advised by Dr. Christopher Ching and Paul Thompson at the Imaging Genetics Center at the University of Southern California, specializing in machine learning and deep learning applications for neuroimaging. Her research focuses on surface-based structural MRI analysis to examine morphological changes in gray and white matter, particularly in the context of neurodegenerative diseases and neuropsychiatric disorders. Her work aims to enhance the accuracy of predictions related to diagnosis, prognosis, and treatment response. Really excited to have you here today, and I'll go ahead and hand it over to you.
You see my screen? Okay. Hi, everyone. Thank you so much for a nice introduction to Cecilia. And today I'm going to talk about the toolkit we've been showing at several conferences, including WCAI. The toolkit name is a 3D brain geometry toolkit for multisite neuroimaging analysis. Before I go into the details of the toolkit, I would like to just talk about what ENIGMA is, even though we briefly talked about it before. But ENIGMA initially started with multisite genome-wise association study, so-called GWAS, and the success of ENIGMA led to the formation of over 50 collaborative working groups, developing standardized protocols and studying a wide range of neuronal conditions. And every pin on the map here represents individual sites. And this international multi-cohort collaboration allows for more accurate testing of the reproducibility of disease effects in participants with neurological and neuropsychiatric conditions, including bipolar disorder. And I would like to note that we formed the collaborative study by revisiting existing brain MRI data sets that were already collected independently, not collecting from scratch. As I mentioned earlier, ENIGMA has been making efforts in standardizing method in different neuroimaging modalities. Like T1-weighted MRI, we did free surfer and longitudinal analysis, and also diffusion MRI and resting states. There has been a lot of works in that modalities. And one of the example is subcritical brain shape analysis pipeline. And today we are going to talk about the standardized way of doing it. Before I talk about the shape analysis in detail, I would like to talk about why we care about shape analysis first. Shape analysis has been known for revealing more fine-grained alterations compared to volumetric analysis. And to show the example, I took ENIGMA schizophrenia working group samples of the readers here. This is gross volumetric analysis for subcortical brain structures. And here, coherency is the statistical metrics that shows which brain structure has larger volume or small volume in schizophrenia patients compared to healthy controls. So negative value indicates smaller volume in schizophrenia, and larger volume positive value indicates larger volume in schizophrenia. And shape analysis result would look like this. It's more high resolution, detailed difference in schizophrenia compared to healthy control. And here, statistical metric was beta coefficient, but you can just focus on if it's negative or positive to see the direction. So hotter color indicates higher thickness, and cooler color indicates lower thickness in schizophrenia compared to healthy control. And I will take hippocampus as an example here. Hippocampus in subcortical gross volumetric analysis, you can only see the one-dimensional changes. That means smaller volume of hippocampus was observed in schizophrenia. But if you take out the shape analysis results from hippocampus, then you can see most of the region has lower thickness in schizophrenia, but also some of the region has higher thickness. So you can see more detailed changes. And putamen, for example, as well, larger volume, only one-dimensional changes in volumetric analysis, but in shape analysis, you can see some of the region has lower thickness. So this is what we would like to see. And from this detailed subfield variation, we can examine neural connections or brain functions related to the altered subfields in neuronal conditions we are interested in. So that's what we want to do through shape analysis. And I would like to acknowledge that many shape toolkit analysis for the analysis exist. SPAM PDM, CERT, LDD, MM. These are all cool toolkits, but today we would like to focus on ENIGMA shape analysis pipeline. So this pipeline was originally developed by Dr. Bruce Goodman, who is located now at IIT, but he worked on this project when he was in USC. And it starts with free surface subcortical segmentation and construct the mesh for those segmented 7 bilateral subcortical volume of interest. And the toolkit register mesh to ENIGMA subcortical shape template using medial demons approach. And later on, we calculate 2 different measures. One is radial distance, and the other one is surface Jacobian. Let's focus on radial distance first. To calculate radial distance, we derive medial curve first. Medial curve is right here, that across the subcortical structure. And to calculate the radial distance, we calculate the distance between each vertices to this medial curve. And it can be interpreted as thickness, like we do in a cortical analysis. And for the surface Jacobian, is calculated by estimating Jacobian determinant to map average template to the surface of subjects. And it represents the surface dilation ratio between the template and the subjects. And in other words, it's a nonlinear warping to see the amount of how much you sketch or expand to make the same shape from template to the individual mesh. So it can be interpreted as surface area in the later part. So this ENIGMA shape pipeline has been applied to a range of brain conditions in neuroimaging researchers, especially ENIGMA working groups. So we would like to talk about the ongoing works based on this existing data later on. And this is the outline of the toolkit that I'm gonna present today. I would like to talk about the common challenges in multi-site neuroimaging analysis first, and then introduce how the brain geometry toolkit address these challenges. And I would like to show the application of this toolkit in large-scale multi-site samples from the ENIGMA bipolar group. Here we go. And this is how the pipeline look like in the toolkit. It starts with ENIGMA shape pipeline first. And because a lot of people run shape analysis from a worldwide range institution, we would like to address some of the challenges in brain geometry data, which are multi-site data standardization, fast and estimation of statistical analysis, and machine learning analysis and visualizations. The first challenge in large scale neuroimaging is combining data from different sites and scanners. If we combining data from different scanners, ABC, for example, here, then we get the scan and then calculate the brain mesh and then get the features of the shape of the subcortical brain. And when we plot the distribution of these features, we get skewed distribution across the sites. But this is what we don't want to get because this is biological non-relevant features. So what we would like to see is more homogeneous distribution in brain features. So to do so in traditional way, linear mixed effects modeling, so-called LME, has been used to address this issue. And this is statistical modeling. So it includes statistical analysis procedures, but also a lot of neuroimaging researchers has run linear combat or combat GAN for data standardization harmonic techniques. And then later on, we run additional linear statistical analysis. But I will talk about LME first, and then we will go into deeper what combat and combat GAN is. So linear mixed effects model is you have linear model for fixed effects and error term, but also on top of that, we have random effects to model sites. So for this linear model, we model individual vertex-wise features using this single independent linear model. So we can do statistical analysis by controlling the site effects here. But for the linear combat, we residue out the additive side effects and multiplicative scatter effects first. But I will just go into how this combat was made. So the combat model was originally developed for gene expression microarray data, and it was reformulated for neuroimaging case by JP 14's paper. And it poses a unique linear model at each feature. And same as well for this, our shape analysis case, it's vertex-wise feature. And it makes assumption that scanner or sites have both an additive side effects and multiplicative effects on the data. And the model assumes that the expected values of imaging features can be modeled as a linear combination of the biological variables and the side effects. And biological variable here was modeled with average value for feature Y and the side effects, and whose error term is modulated by multiplicative scanner effects. And the algorithm uses empirical base to improve the estimation of the model parameters in small sample size studies. And after we standardize the data, we take out the data and then we run linear regression to see the statistical difference in groups. And for convex GAN, it looks similar, but only difference is this part. This part is nonlinear covariate effects modeling using generalized additive model. And usually neuroimaging researcher model nonlinear age trends in ROI volumes informed by the data. And same as well here, we take out all the vertex-wise features. And then we run linear regression on standardized data from convex GAN. Another challenge we would like to see here is computational burden for large scale studies. If we take out one standard space brain scan, it has usually over 500,000 voxels. And if we say fitting a model took 0.5 seconds, then total time would be about 3 days. And say 30 seconds is going to take, then total time would be 190 days. And sometimes it can take several hours to fit even a single model. So then now it becomes more problematic. We cannot really run these models in time. Dr. Pravesh Parekh from JCVI in San Diego and their team developed this cool toolkit called Fast and Efficient Mixed Effects Algorithm, so-called FEMA. And they applied this to vertex-wise analysis for critical data and connectome-wide analysis and voxel-wise analysis. And for their case, they modeled longitudinal effects and family and genetic effects. So a lot of modeling is involved. So using standard LME, it took almost hours to days of time. But when they ran FEMA on those cases, it only took seconds to minutes. So it was very significantly faster. But I will show how it looked like in the ENIGMA case in the later part. But for the ENIGMA case, we go over all the detailed quality control procedures. So we cannot make use of just running FEMA on one iteration because we need to run ROI-wise iteration. So it can take more time because of that reason, but still it was very faster compared to standard LME. I will talk about in the later. And using this FEMA in our toolkit, linear regression is included. So we can now use it through the toolkit. For the linear mixed effects modeling, Dr. Pravesh and I are doing some works to stabilize the results. So stay tuned. We are going to release so soon. So then now everyone would wonder why FEMA is so fast. So the key here is we make use of the sparsity in the random effects design metrics. So random effects design metrics usually have lots of sparsity is going on, but where the sparsity exists, the FEMA skips the computations from that parts, so it can boost the speed in that aspect. And to do so, they use method of moments and regression estimator for random effects and binning strategy for multi-outcome measures. And also vectorized coding with scalable code design was used for this process. And now I would like to talk about AI functionality of the toolkit. Before I go over the details of AI functionality, I would like to talk about why we apply AI to brain features, especially subcortical shape features today. So let's say we take these shape features from brain mesh and then put into artificial neural network. Then we can predict diagnosis, treatment, and illness severity. So for this aspect, in this aspect, clinician maybe get advice from this model by building a classifier to classify disorders. So they even before look at the scans, they can see, oh, okay, this brain scan maybe have a potential disorder, so it's worth to look at it. But most of researcher focusing on neuroimaging studies is aiming to find neuroimaging markers for disorders. So to do so, it's very important to have interpretability from AI functionality. So Brain Geometry Toolkit provides that using Logic TV-L1. This would look complicated, but I will dissect one by one to understand quickly. So Logic is coming from logistic function or logistic regression. So we build logistic regression model using this. We take out n vertices features first to build a logistic regression model. We make one-dimensional features using these n features. And then we also prepare the one-dimensional weights to do linear combination with the feature. And then after we get the result from linear combination, now we put in the register function and we can get the probability of a disorder. And when we optimize the weights here, what we want to do is we want to improve the accuracy of predicting a disorder in this model case. But not only that, we would like to add two different regularization techniques here so that we can get interpretability from AI. One regularization technique is L1. You guys may be very familiar with this, but this L1 regularization is especially smooth version so that we can get very smooth sparsity here. And as I mentioned, it encourages sparsity in this L1 regularization. And another regularization is TV, total variation regularization. And this regularization is imaging aspect of regularization, so it encourages spatial coherence. So we can get more coherent weights map when we take out the weights. So now after we train this model, we can take out the weights and then we can visualize on brain mesh so that we can see which brain region was highly focused on for this specific task we are doing. And Brain Geometry Toolkit also supports a range of visualization functions. When we visualize the subcortical brain here in the original view, so-called native anatomic spatial configuration, you can see the brain structures are very interconnected. So it's hard to see the occlusive part. So what our USC team developed was unpacked view. This is very beneficial because we can see all the brain structures all at once. But the problem is we cannot see which brain region was originally located. So we need to go back to the reference again. So what we developed here is interactive visualization. If you scan this QR code, then you can interact with this figure to see all the occluded brain regions. And then in an anatomical configuration, you can easily investigate all the brain regions. And now I would like to show the application of the toolkit in large-scale multi-site samples from ENIGMA Bipolar Group. From ENIGMA Bipolar Group, 21 sites participated in this project. Age range was very diverse, and male ratio was indicated for 2%, and bipolar disorder and healthy control samples were looking like this. So in total, it was over 3,000 samples. And as I showed you earlier, I would like to go over why shape analysis is beneficial compared to volumetric analysis in bipolar disorder case 2. This is subcortical brain analysis results using volumetric analysis. And you can see here, what we can see is one-dimensional direction. Here, same as well, Cohen's d was used to investigate the direction. So hippocampus and thalamus was smaller in bipolar disorder compared to healthy control, and other regions also one-dimensional changes. But brain shape analysis had more high resolution changes was captured. Here also Cohen's d was used, hotter color, higher thickness, and cooler color, lower thickness. And because the toolkit supports other data standardization technique like linear combat and combat game, so we were able to obtain all the results from using different data standardized techniques. So let's take out putamen first here as an example, and from shape as well. So here, from volumetric analysis, show putamen volume was larger in bipolar cases compared to healthy control. But when we take out the putamen from shape analysis, you can see some of the regions were having higher thickness, but actually some of the regions had lower thickness in bipolar disorder cases. So as you can see, shape analysis has benefits to see the high resolution of the changes in subcortical brain structures. And as I mentioned earlier, larger scale statistical modeling encounters high computational demands. And for the ENIGMA bipolar working group case, also it had larger samples, over 3,000, and also shape analysis has over 27,000 features. So it requires very high computational demands. So when we applied FEMA, we obtained 16 times faster speed in the statistical analysis. So it was from minutes to hour to seconds to minutes. So it was very beneficial using FEMA. And then now I'm going to talk about AI functionality part. But before I go into that, I would like to talk about what ENIGMA has been doing in terms of machine learning, and also I will show the results and the goals here. So ENIGMA has been focusing on critical thickness and subcritical volumes like this from different ENIGMA working group. And what we would like to do is compare models trained on these features, regional measures, with model trained shape features. So here, actually from bipolar working group, there was a machine learning study using free surface measures and subcritical volumes. And when they implemented this study, they used support vector machine to classify bipolar disorder from control diagnostic classification. And the accuracy of the classification ranged from below the chance to 65.23. So you can see bipolar disorder classification task is very difficult classification. And when we investigated brain features driving prediction both biologically relevant and consistent across study samples, we found that anti-epileptic treatment was associated with more accurate bipolar disorder versus control classification. So this was the study using free surface measures and subcortical volumes. But now I would like to talk about the case using shape features. So I will go over setting first. We used Logit TV-L1 models, and we trained to predict bipolar disorder diagnosis and lithium and anti-epileptic treatment at the time of scan, and history of psychosis and bipolar disorder age of onset prior to 21 years old versus after age 21 using vertex-wise thickness only. And optimal total variant and L1 regularization parameters were searched by grade search using 10-fold cross validation. especially the balanced accuracy was used to evaluate which parameter is optimal. And linear SVM models were trained on gross volumetric measures of subcortical brain volumes specifically, and it was compared with shape feature model. And for all the clinical test cases, the models trained on volumetric measures had non-significant result. And here, the non-significant result means we ran multiple comparison test using balanced accuracy from 10-fold cross validation with FDR correction. So when we obtained 10 different balanced accuracy, and we did t-test on that, and it turns out volumetric measures model didn't have significant results but for models trained on shape features, had significant results, except for one task, which was history of psychosis prediction. And as I mentioned earlier, we can also investigate the weights map that drove performance of predicting each cases. And here's the bipolar disorder diagnosis case. And as I mentioned earlier, total variance and L1 parameter is chosen based on the performance. So we cannot guarantee if we fix the parameter, then we cannot guarantee if the model is going to be having higher performance in the task. So here we got lower parameter values for two different regularization. That means lower TV means higher coherence, and lower L1 means smaller sparsity. So we are able to get more relatively interpretable weights map here. And if you remember from the previous slides, it has similar overlapping regions with statistical results. And here is lithium prediction weights map. And as we expected, thicker thalamus and hippocampus is here and thalamus is here. We got the expected results from this weights map. And also because we had higher coherence and similar sparsity from this parameter, we were able to interpret more intuitively. But for the anti-epileptic prediction case, it's very hard case. So model was struggling with to find good performance, and also total variant was chosen with higher value. That means smaller coherence. So the weights map had very smaller coherence looking. And because of that, without arbitrary threshold, we are not able to interpret this weights map. Now I'm going to talk about ongoing and future works. And as I mentioned earlier, one of the ongoing work is cross-disorder shape analysis. ENIGMA working group has ran this shape pipeline on their data. And now what we would like to do is to make use of existing data for these shape structures and then do cross-disorder shape analysis. In this project, we are aiming to answer the following research questions. One is what are the overlapping and distinct shape patterns across disorders? Second, can we train models to distinguish disorders? And finally, can AI model trained on shape features boost the performance for clinical tasks, such as predicting treatments, severity, and etc.? And on the right side, we have the table of the sample size from the different working groups we are reaching out to. And in the results, we will have very impressive sample size here. And this is future work for the toolkit. First step we would like to do is to integrate cortical surface analysis protocol and add other machine learning interpretable AI models to improve performance of classifications. And I would like to take a moment to acknowledge that ENIGMA Bipolar working group participants has contributed their data that they've been putting their time and efforts and also helping out the drafting manuscript. And also my amazing mentors, Dr. Ching and Thompson, and also wonderful colleague, Melody. And if you have any questions, please let me know. And if you can scan this QR code, you can check out the recent ENIGMA bipolar works and paper for the toolkit and my link to looks like to run this visualization parts. I will start with first. So here, what you need is statistical results, CSV file, and I will upload how it looks like, what should be ready for that, because I will upload the template to run this analysis. So you can just put the results to the template, and then you can read using pandas. And then after you read it, now what you need to do is import this toolkit. And then my toolkit also has rendering part, which means you have the statistical analysis results and then you render to the shape. So rendering part is done by the function load results. And then now you have mask beta dict. And also, I forgot to specify all the details here, but here you can put ROI IDs. So you can put which structure you will visualize. So 10 to 58. This is indicated in ENIGMA tutorial for shape analysis pipeline, but you can put all the numbers that indicates different structures. And for text counts and metrics here, I focused on thickness. And correction method is because my toolkit supports different correction for multiple comparison. So you can put different corrections to visualize different correction-based results. And threshold is the p-value you're gonna threshold. So then after that, what you get here is masked beta or cohesity values. And mask means because we would like to see only significant result like this. So we wanna threshold the regions that doesn't have significant results so that it can make gray area like this. So I will start with interactive figure first. Interactive figure, what you need to do is you call pull-up brain first, and then you can specify the parameter interactive true. And then you can specify all the different things, but these are not necessary. So I will go one by one. So to visualize the subcortical brain, what you need is you need ENIGMA subcortical brain template. So this is what I call out from ENIGMA template, but I will also upload this to the GitHub so you can specify the path to the further after you download this atlas. And then again, specify the RI meshes. And then you can also label the cursors. So when you interact here, the cursor says it's even though it's white, but left putamen. So you can specify all the different shapes in a different labels. And then you can also specify the value ranges because bipolar disorder case has maximum 25.25 coherency. So I specify this. And also you can customize the color bar, but I made this color bar because I wanted to non-significant results as a gray. So you can also just make it white, whatever you want, by calling this function. And then put the masked beta or cohensity here, and then save the path. Then what you get here is, I think it will take a little bit of time. Oh yeah. So you can get interactive figure like this. And also, as I show here, you can also get HTML file. So here's the path. So when you get HTML file, the benefit of this is you can upload to the GitHub, and then you can share with other people, or you can even put in the paper so that readers can also access to it. And then in the same function, if you specify interactive as a false, then now you can plot all different static plots. And here there are 6 modes, and modes 1 and -1 is for exploded, in other words, unpacked view of the figure. So only difference here is because we don't need to specify all this cursor. So you can just exclude all this part, but you can only specify what's necessary. And then as a result, mode 1, you can get this unpacked view of the figure. And dorsal and ventral is supported with mode 1 and minus. And for the original view, we have 4 different views. One is top and bottom and front and back. So for that result, we have all these different views we can get. And you can also if you edit the code in the plot brain, you can also rotate as much as you want, and you can get different views as well. So here's what we provide in the visualization components. And other components will be releasing once we organize everything to the official GitHub page.